Some human proteins, like insulin, need to be produced in large volumes to be used as medical treatments. One way of doing this is to transfer the gene that codes for the protein into a host cell that can be easily cultured. How do scientists do this? To understand this, we need to explain how DNA fragments can be inserted into a vector, describe how host cells are transformed, describe how marker genes are used. Once the gene for a protein like insulin has been identified, it needs to be isolated. Restriction endonucleases are used to cut the desired section of DNA from longer strands. Some of these cut straight across, but others produce a staggered cut and leave a few nucleotides of single-stranded DNA. This is called a sticky end. A vector is used to transport the DNA fragment into a host cell. A plasmid can be used as a vector if the host cells are bacteria. The plasmid is cut with the same restriction endonuclease. This leaves the plasmid with sticky ends that are complementary to those on the gene. The opened plasmids are mixed with DNA fragments, and the complementary sticky ends match up. The sugar phosphate backbones of the two complementary sections of DNA are joined by an enzyme called DNA ligase. Some plasmids are closed without incorporating the desired gene. The vector containing the gene must be introduced into the host cell. This process is called transformation. The cell membranes of bacteria are made more permeable by electroporation. The bacteria are then mixed with the plasmids, allowing the plasmids to move into the cytoplasm of the bacterial cells. Not all host cells will take up the vector. To identify bacteria that have taken up the plasmid, the bacteria are exposed to an antibiotic that the plasmid carries resistance to. Only bacteria that have taken up the plasmid will survive. Not all vectors that are taken up contain the gene to be cloned. Marker genes are used to identify the cells that have taken up vectors containing the desired gene. The gene that is being cloned is inserted into the middle of a marker gene. Green fluorescent protein, GFP, is a protein produced by jellyfish. The gene that codes for GFP is used as a marker gene. After transformation, host cells are viewed under a microscope. Any cells that fluoresce have taken up a plasmid without the desired gene. Cells that do not fluoresce have taken up the plasmid containing the gene to be cloned. The host cells that have taken up the plasmid are cultured to produce the protein coded by the gene in large quantities. The required gene is identified and isolated using restriction endonucleases. The isolated gene is inserted into a vector, such as a plasmid, using the same restriction endonuclease to cut the plasmid and DNA ligase to join the strands together. The vector is introduced into a suitable host cell in a process called transformation. Markers are used to identify the host cells that have taken up vectors containing the gene to be cloned. These cells are then cultured to produce the gene or protein in large quantities.